Welcome. Today we are talking about a virus that is a true master of hide and seek, herpes simplex. We're going to break down all the high-yield facts you absolutely need to know, from its super clever life cycle all the way to the critical treatments. So let's just dive right in.
All right, so here's our game plan. First, we'll get a profile on the virus itself. Then, we'll trace its journey through the body, see what it looks like in the clinic, figure out how to diagnose it in the lab, and finally, we'll cover how to treat and prevent it.
Okay, first things first, let's get to know our culprit. What exactly is herpes simplex virus?
So HSV is part of this big family called Herpes viridae. They're these large, enveloped DNA viruses. But here's their defining feature. The one thing you absolutely have to remember for your exams. They set up shop for life. They establish these lifelong latent infections. Once this virus is in your system, it really never leaves. So what makes this virus tick? Well, for one, it replicates super fast, and it's cytolytic. That's a fancy word that just means it literally destroys the host cells it infects, and that's what causes those classic blisters. But the absolute most crucial feature, its signature move, is its ability to establish latency, basically to go dormant, and it does this specifically inside our neurons.
Now we split HSV into two main players, HSV1 and HSV2. A simple way to think about it is that HSV1 is usually spread through oral contact, saliva, and causes lesions above the waist. Think cold sores. HSV2, on the other hand, is mostly sexually transmitted and causes lesions below the waist. Now, can there be overlap? Yes, absolutely. But this waist rule is a really solid rule of thumb for the clinic.
Okay, so now we know who the virus is. Let's dig into its pathogenesis. How does it actually work? Understanding its journey through the body is the key to understanding all the different diseases it can cause.
You can think of its life cycle in four main steps. First, the primary infection. The virus gets in through a little break in the skin and starts making copies of itself right there. Second, it invades the local nerve endings and travels up the nerve. Third, it reaches the dorsal root ganglia and goes into hiding, establishing that lifelong latency. And finally, reactivation. Something triggers it, and it travels right back down that same nerve to cause trouble all over again.
Now, this is a super high-yield point. Where the virus hides is incredibly important. HSV1 loves to hide out in the trigeminal ganglia, which, as you know, supplies the face. Meanwhile, HSV2 prefers the sacral ganglia, which supplies the genital area. So you see, its hiding spot directly predicts where those recurrent outbreaks are going to pop up. Makes sense, right?
So, the virus is just sleeping in our neurons. What wakes it up? Well, a few things can act as an alarm clock. A fever is a classic one. That's why we call them fever blisters. But also things like emotional or physical stress, an injury to the nerve, or even something as simple as getting too much sun can trigger a reactivation.
All right, let's move into the clinic. What does an HSV infection actually look like in a patient? The short answer is, it can look like a lot of different things.
And that's because HSV is what we call a pluripotent pathogen. It's a real jack-of-all-trades when it comes to causing disease. It's so versatile that it can cause this huge spectrum of problems, from a simple little cold sore all the way to a life-threatening brain infection. Let's go through some of the big ones.
So the most common things you're going to see are lesions on the skin and mucous membranes. For HSV1, that's your classic gingivostomatitis or cold sore. For HSV2, you're looking at painful genital ulcers. But then you have these other interesting presentations like herpetic whitlow on the fingers. You see this in healthcare workers. Or herpes gladiatorum, which wrestlers can get from all that skin-to-skin contact.
Okay, now listen up, because this is a huge one for your exams. When HSV gets in the eye, it can cause something called keratoconjunctivitis. And the classic must-know sign is the dendritic ulcer. If you put a special stain on the cornea, you'll see this amazing zigzag branching pattern that looks like a tree branch. It's a dead giveaway for HSV, and you do not want to miss it.
Now, things get really, really serious when HSV gets into the central nervous system. It can be devastating, and it's critical to know the difference between the two main ways it presents. HSV1 is the #1 cause of sporadic viral encephalitis, and it has this classic tendency to affect the temporal lobe. HSV2, on the other hand, is more often the culprit in meningitis, and it can even cause this specific recurrent type called Molleret's meningitis. Neonatal herpes is an incredibly serious topic. Look at this chart. The risk of passing the virus to the baby is about 10 times higher if the mother gets her first HSV infection during pregnancy compared to if she just has a recurrent outbreak.
Why? Because with a primary infection, she hasn't had time to make any protective antibodies to pass along to the baby. It's a really crucial distinction.
Okay, so you see a patient. The signs are pointing to herpes. But how do you confirm it? How do you know for sure? Let's talk about the lab.
All right, here's the question you have to be ready for. What exactly are you looking for in those lab reports that's going to scream herpes simplex virus and lock in your diagnosis?
So here are our main tools. The T-Zank smear is quick and easy, but is not specific. It can also be positive with the chickenpox virus. Virus culture is specific, but man, it's slow. Serology is great for telling the difference between HSV1 and HSV2, but the one you absolutely need to burn into your brain is PCR. For any CNS infection, doing a PCR on the spinal fluid is, without a doubt, the gold standard. It's fast, it's sensitive, it's specific, it's the best we've got.
And let's just zoom in on that Zank smear for a second, because this is a classic histology question. Under the microscope, you're looking for these huge, multinucleated giant cells. You might also spot these things called Cowdrey type A inclusion bodies inside the nucleus. That's a classic hallmark finding for the whole Herpesvirus family.
And finally, let's get to the most practical part. What can we actually do about this? How do we treat an active infection, and how do we stop it from spreading?
The drug you need to know is acyclovir. This is the cornerstone of treatment, the absolute go-to. And how it works is actually pretty cool. It very specifically targets the viral DNA polymerase. That's the enzyme the virus uses to copy its own DNA. So acyclovir just puts a stop to that whole process. No DNA replication means no new virus. It's really elegant.
And I want to show you why treatment is so unbelievably critical. Just look at this number. The mortality rate for a newborn with herpes, if it goes untreated, is a staggering 65%. That is a chilling statistic, and it really drives home why we have to diagnose it quickly and start acyclovir immediately in these babies.
So what about prevention? Well, as of now, there's no vaccine. To prevent sexual transmission, condoms help, and people with frequent outbreaks can take suppressive antiviral therapy. But the most important, life-saving intervention is for newborns. If a mother has active genital lesions when she goes into labor, the recommendation is an elective C-section. This physically bypasses the site of infection and can save the baby's life.
And that's a wrap. That is our high-yield breakdown of herpes simplex virus. We've covered its basic biology, what it looks like in patients, how to diagnose it, and how to manage it. If you found this explainer helpful for your studies, do me a favor and hit that like button, leave a comment, and subscribe for more content just like this.